So today is going to be a very, very busy day. I need to look after my stem cells, start generating organoids. I've got a few introduction meetings, and I need to slice my brain on the microtome. I know it doesn't sound like a lot, but trust me, it'll take the whole day. So let's go. So before I even step into the lab, I spend about 30 minutes every day planning. I write everything down I need to get done and a few things I want to get done, if I have time. Seeing it all laid out in front of me helps me stay calm and focused. Otherwise, everything will just blur into one big to-do list in my head. Planning my day keeps my schedule realistic, and it also helps me prioritize what really matters, and it stops my day from snowballing into pure chaos. And I also time block for breaks too. My first stop is tissue culture, and my stem cells are so needy. They need feeding, cleaning, and just the right environment to stay happy. If you've never worked with stem cells before, think of it as babysitting thousands of tiny drama queens. They demand the perfect temperature, the perfect light, and constant attention. They're temperature sensitive, light sensitive, and have absolutely zero tolerance for mistakes. I spend a lot of time making sure they're healthy and happy and ready to generate into organoids. One contaminated plate and weeks of work can disappear. So yeah, it can be a little stressful. But on the other hand, it's also kind of magical to think that these stem cells can become almost any cell in the body. From neurons to muscles or liver cells, and even grow into complex structures called organoids that mimic mini organs. And this makes all the pipetting feel worth it. Next up is slicing brain tissue. It sounds intense, but honestly, it's not that bad. The slicing itself isn't too difficult, but it's the setting up of the microtome that may take some patience. Every tiny adjustment, the blade angle, the tissue temperature, even how you handle the sample, can make the difference between a perfect slice and a crumpled mess. These slices are then ready to be stained and imaged to later reveal the cell type, the neural connections, and the patterns that tell us about development and disease. Towards the end of the day, I like to slow things down and switch gears. This is where I catch up on reading or writing notes, and I also find it useful to write down what I've done that day.